Good evening. I'm Yvonne, Staff for Science for the Public, and I welcome you to Contemporary Science Issues and Innovations. Tonight's guest discusses the study of plants that have medicinal properties and how those properties can be used for the development of modern medications. Dr. Jing-Ke Weng is Associate Professor of Biology at MIT and a member of the Whitehead Institute. His innovative research is drawing a lot of attention, and he's received numerous awards. The Weng Lab combines analysis of plant genes, called genomics, with genetic engineering to determine potential medical value for diverse medical problems, ranging from pain to conditions like diabetes and Parkinson's. Dr. Weng will explain how his lab analyzes and simulates the medicinal properties of various plants, and also how those properties evolve. And we'll learn about ancient uses of plants as well. It's a great pleasure to welcome Dr. Jing-Ke Weng. Welcome, sir.
Thank you. Happy to be here.
And I'll ask if you could give us a little background about medicinal plants that have been used for centuries, apparently.
Yeah, so we have a lot of plants growing on the planet. So reportedly, there are over 400,000 species. We can recognize a few around us. Mm-hmm. And throughout the human history, humans have been using many plants for treating disease. Mm-hmm. And now we have modern scientific methods. Now we can uncover the mechanism why these plants are useful and what are the molecules that are playing these roles. Hopefully, we can develop new medicines starting from that point.
I see. Now, in terms of those, the use in, well, through the centuries of plants and combinations of plants without a scientific method there, how effective are they in general?
Yeah, so we have
in the traditional
way. Humans originated from Africa, and we started as hunter-gatherers.
Yeah.
I think most of the diet for ancient humans were plants, maybe occasionally animals, but we did experiment things that are edible. So inevitably, through the process, people would discover things that have accidental medicinal properties.
Uh-huh.
People were getting sick, and by eating certain plants, people get better. So before language, things were just passed through tradition, generation to generation. And then when we have written language, that tradition can be written down and becoming summarized and then passed down to further generations.
I see.
So, and this process, as you can imagine, could happen in multiple places around the world before different cultures can exchange knowledge. So I was born and raised in China, so I knew more about Chinese traditional medicine. So it started thousands of years ago. I think the first text was compiled 2000 BC. So it's called a Divine Farmer's Materia Medica.
I see.
So in that book, it was basically summarizing about 120 medicinal plants known to people at that time. So things become added to the traditional medical text over time. So there are very renowned medical doctors throughout history, and they have spent their time practicing, experimenting, summarizing what has done before them, and now we are faced with this very rich body of text. And in other cultures, for example, in South America, it's more passed down through oral traditions. So we have shamans from Amazon rainforests,
right,
and they know what plants to use for certain illnesses. And the people can still visit these places and learn about these traditions. So I would say it's a wonderful resources to discover medical properties of plants.
Nice of nature to provide that, right? There are plants that are visited or used by animals as well, so they don't write books about it or anything, but this is throughout nature, it seems as though these properties are known.
Yeah, so to me as a scientist, I really kind of see a very much larger time frame for the evolution of the
Yeah,
Earth.
yeah.
So I mentioned the origin of modern humans,
Yes.
Homo sapiens,
Yes.
really came about in the last 100,000 years on the planet. That's a very, very short period of time considering the lifetime of the Earth. So plants or biosynthetic or photosynthetic organisms arose much earlier.
Yeah.
And most of the plants we're very familiar with, or the source of medicinal plants, are land plants. So there was a very important event that occurred on the planet, which is called the land colonization by plants. So most of the plants lived in the water.
Yeah.
About 500 million years ago, the first land plants emerged. So they really have to deal with a lot of the stresses which are only present on the terrestrial environment. Like they have to support themselves. It used to be they're supported by floating. They have to defend against a very dry environment, the drought environment. The sun.
Dark, yeah.
And once they succeeded to be able to live on land, there are also co-evolving bacteria, fungi, and other animals which have to feed on plants to survive. So they really have to defend themselves. As we know that plants are so-called sessile organisms. So once they're rooted, they can't move around. So the main strategy for them to survive is to become very good chemists. So over hundreds of million years, they really elaborate their metabolic systems, which as humans, we also have those systems to synthesize fundamental building blocks and energy molecules. But for plants, in addition to those requirements, they make a lot of additional molecules to defend themselves.
I see.
Some contribute to their taste, their color, and many become drug-like. It's just really to toxify anything that would eat them.
I see. Or to build some other kind of relationship. But this is a very important point, that this is an evolutionary process. It doesn't start on day one and not change, but they adapt to other organisms and conditions, I guess.
Right. It's a remarkable long-term process that has taken hundreds of million years and involving many species and their environmental niches.
Yes.
Specific species also diverge. We have a huge diversity of plant species and the insects and animals interacting with them.
Yeah.
And as the end result of this process, arms race, you have very effective compounds that can protect specific plants against any herbivores or pathogens that would harm them.
Right.
So for humans, we really arose much later in time. By accident, we stumbled upon those plants and discovered the medicinal properties.
Right. Could you give us a few examples, just before we get into your lab?
Sure, yeah.
A few examples of plants that are familiar. I was thinking of, like, is it the willow, some willow that was used for aspirin, and I don't know if there was one for like malaria. I don't know these different.
Yeah, you're already very, very well versed in the field.
Oh, there we are.
the willow plant, it's a tree, and people in ancient time has discovered this salicylic acid derived molecule present in the tree bark. If you eat it, you can ease pain. And later on, people found aspirin. Essentially, it's the same molecule. Another plant, opium poppy, people know a lot.
Right.
Yeah,
they do know that one, right?
Now we have opioid crisis.
Yes.
But we have to admit morphine, which is the most abundant and effective compound from the plant, is the first drug approved by FDA.
Oh, how about that? No, I didn't know that.
Yeah, it is such an effective compound to ease pain. And these days we use other types of opioids, which are all modified from the initial molecule, because the first one is just too effective. People get addiction very easily. Yeah. By toning this down, by changing the molecule, you can get better drugs.
And that's what you're working on too. And I want to come back just before we get into that. There are plants that are effective for a variety of diseases, parasites and things. How about viruses?
Yeah, so viruses, bacteria
effective,
and fungal pathogens. These are
okay.
microbes that the plants have to deal with all the time.
Yes.
We're still in the process understanding what are the molecules that plants
Oh,
make
yeah,
to defend
yeah.
them. But I would also add to that, there's a lot of value of studying this because we now have huge issue about antibiotic resistance.
Absolutely.
We're running out of effective antibiotics.
Right.
And most of these actually are produced by bacteria and fungi.
Yes.
Few efforts has been put into identifying plant-derived antibiotics. I have a few colleagues in the field. They found there are tremendous diversity of compounds produced in plants that have very effective antifungal, antibacterial activities. I think this will become a very exciting source for future medicine in this area.
And more resilient to this problem of the antibiotic resistance problem.
Yeah,
So.
there's another point which we found is when you challenge a plant with a virus or a bacteria, oftentimes they come up with a battery of the molecules. It's really a combinatorial approach they throw to these pathogens.
Yes.
And I think single molecule drug oftentimes leads to drug resistance.
I see.
So by knowing how plants come up with different molecules, the pathway these molecules act upon, we can devise new therapeutic strategies for these infection diseases.
Let's go to this directly because this is what your lab is famous for, especially now. And it sounds from what I've read about you that this is really going to be a big field down the line. I didn't realize that the plants are so clever that it's not like it takes centuries for them to devise a new solution. They can do it pretty fast, it sounds like.
Yeah, the way you put it is very poetic. They're clever. They actually don't have a brain. They don't run around. So the existing plants we see today is actually all survivors from very cruel competition. Anything that could not survive pathogen fungal attack dies. So only small numbers of the individuals at any given generation survive. So we're really looking at very remarkable mechanisms that have been selected through very cruel selection, natural selection. So there's another example I want to raise because I think the audience will be interested to hear is broccoli, right? So the cabbage family, including broccoli, makes a very specific class of molecules known as glucosinolates. And they're very spicy. So the extreme case is wasabi. If you eat wasabi,
yes, indeed.
the horseradish, that's the pungent taste, which is very effective deterrent, as you can imagine. So the nice thing is these molecules are very toxic to anything that eats it, but may also toxify the plants themselves. So plants actually devised a very clever mechanism. So they only make this molecule all the way to the last step. The last step, it has to be catalyzed by an enzyme that's separated from the precursor. So it's only when the plant is damaged by insect chewing, the final product is made.
That's very clever.
So that plants have made this in a way so that the healthy tissue would never have to deal with this problem. And the only effective compounds are made when the plants are damaged.
Attacked or something like that.
That's right, yeah.
That's very clever. And you can't help but avoid the word clever. But
right, and these molecules are...
They're just reacting.
Yeah, and these molecules are so interesting. People are exploring the anti-cancer activities of these molecules.
Yes, and on that, I had the impression that, so I still don't know whether there are plant medications that are effective against viruses as opposed to bacterial or fungal or other kinds of diseases.
Yeah, viruses
viruses
is
are another thing.
uncharted territory.
Okay.
I'm sure there are a lot of molecules that plants throw against
Okay.
viruses because there are a lot of plant viruses.
Yes.
But it's currently under study.
Okay, all right, because that will be very interesting, given that these viral diseases are making quite a comeback, like measles, for
That's
instance,
right.
polio
Yeah.
in some places, things like that. But in any case, let's go to your lab.
Okay, sure. Yeah.
Okay, you've pointed out that you can now understand these mechanisms, you can really analyze these mechanisms. What gives you that ability?
Yeah. So people study the medicinal property of plants for decades. I think I mentioned the case of opium poppy. So it's really the beginning of the 20th century. And in the 1960s and 70s, NIH actually organized a very good effort going to the tropical forests and systematically harness plants, isolating compounds, testing their activities against cancer. So a lot of great molecules have been identified, have very interesting bioactivities, but then afterwards people realized it is very hard to carry from that point on. So that whole effort has generated a few drugs that we still use today, like Taxol, which is a first-line chemotherapy drug.
I see.
But these also become the most expensive drugs to manufacture. Taxol is present in the tree bark of the yew tree. For treating one patient, you need to cut down 3 trees.
Oh, wow.
And these trees takes hundreds of years to grow. So the challenge is really to have a source, a stable supply source for these very effective molecules. And also the way the efficiency these molecules are discovered have also been very slow. So it takes many researchers many years to just discover one.
I see.
So what has changed in the last couple of years is the emergence of genomics and many other so-called omics type of studies. So just speaking about genomics, now we can spend about $400 to sequence the whole entire human genome.
This happened so fast.
So fast. It's exponential.
It was so expensive and just huge work, and it's very short time ago, really.
Yeah. At the beginning of 2000s, we spent $3 billion to sequence the human genome. Now it's about $400. So the speed of discovery has been really promoted by the development of technology.
Right.
So we are applying those genomics tools to study plants.
Okay.
So at this point, the whole research community have sequenced the genome and sometimes transcriptome, which are the genes expressed in any plants, for more than thousands of the plants. Many of these are medicinal plants. So that effort has revealed the whole tool sets that the plants have evolved over hundreds of million years that they use to make these interesting molecules.
On that, since you're able to do this, and there's a huge range of plants and stuff, is there like a subset that you kind of target in genetically plant to plant to plant? Is there something, a characteristic?
Yeah, so in the past 30 years, people have been focused on model plants, including a plant called Arabidopsis thaliana. So you can do genetics on it.
Yes.
Or sometimes crops, maize and rice. Few efforts have been focused on medicinal plants because they're so-called dumb model, that there isn't any genetic or genomic resources developed to them. So it's been very hard to study those plants. But now we can quickly sequence them. So that's one component.
Right.
The second component is the ability to profile the chemistry of those plants. Now we have very advanced analytical chemistry methods like mass spec and other advanced technology. So we can really know what the chemical diversity that are present in any of those non-model medicinal plants.
Okay, now I have the impression that you could have, say, a variety of, say, 50 plants and you just want to know what each one can offer, and this genomic approach and your technology to identify it can do that very quickly.
Yeah,
10 years ago, I'm not sure that was easy.
It really happened within the last 10 years. What we do is we focus on a plant with particular medicinal property, oftentimes unknown chemistry, unknown mechanism,
Okay.
but the plant could have been used by a native tribe for doing something for thousands of years.
Right.
And we also know there are plants that are related to this one, but doesn't have that property. So then we do two things.
That's what I want.
We do the comparative study to see what genes are present in this plant of interest. What are the molecules that are made in these plants that are not made in the related species? Through that comparison, then we can identify those genes that probably only evolve in that plant to make the molecule of interest. So we've achieved the genetic basis for this trait and also the molecules that you can further develop into modern therapeutics.
And once you get that kind of information, can I flip back to that Kava plant again? You'll have to straighten me out on this, but reading about what your lab was doing, I also understand now developing the Kava plant is a slow process because it doesn't, you can't breed it easily. Is that true?
Yeah,
but if you
the Kava story is a very exciting and personal one to me.
Oh, really?
I would say currently it's really an uncharted territory. There are so many things we can study. Oftentimes people ask me, how do you pick a project?
Yes.
So I can tell you about how we started the Kava project. About 4 years ago, I attended a seminar, an after-dinner seminar at Harvard Natural History Museum. So at the time, there was an ethnobotanist visiting there from New York Botanical Garden. And he talked about his story going into those Polynesian islands and learned about this plant called the Kava. So it is a plant related to black pepper, but only grows on those tropical islands. And the people there use this for ritual. And it has very distinctive calming effect. So if tribes have conflicts, the tribe leaders would organize those drinking, talking about the problems, and over the night of drinking, then people get very calm.
We should do this for international purposes.
Exactly, right. It's going to be very relevant these days.
Right, I should
and
say.
the problem solved.
Right.
So I heard about this. It's extremely interesting to me, cases like this. And I took out my phone. I searched up this plant. Actually, people have done a lot of chemistry. So they reported the main compound for the plant is called Kava lactones.
Okay.
So the plants not making one actually make a panel. So there are over 20 different Kava lactones that are produced by those plants. Actually, plants coming from different islands have a different profiles.
That's surprising.
Which give people different experience. Some have more calming effects. Some actually keep people a little happy. So as the lab, the postdoc who's working on this has also organized the traditional tasting.
Test everything.
And he has tested multiple brands and really firsthand experience, the effectiveness of this.
I see. That's a lot of fun.
So that's the start for the project.
I see.
So I think,
On that, apparently they're looking at the Kava as a substitute for these opioids. Is that correct?
Right. So
because it's not just calming, but it relieves pain.
Right. So it's a safe, non-addictive alternative for opioids.
Okay.
So it's gaining popularity. But as you mentioned, Kava plants is very hard to cultivate.
Yes, could you explain that?
They only grow on those islands, and the root cutting is used to make this powder, and then you can make a supplement. Although it tastes super bad, it's very hard for quality control. If it gains popularity around the world, there will not be enough production from those islands.
Right.
That's why we wanted really to develop new synthetic biology approaches to produce this from a very sustainable manner.
And is that something that's relatively easy, the synthesizing, and I'm thinking about how difficult it was in the early days with the antibiotic, a penicillin, that it was very hard to get enough.
Yeah, although chemists have come a long way to improve their techniques, but when it comes to those natural products from plants, it is still almost nearly impossible to make them synthetically. So really, we have to learn how plants did it.
Yeah.
They use their genes to assemble so-called metabolic pathways, step by step, starting from common precursor, could be an amino acid that we all have, and through specific transformations coming to those very specific molecules that are only present in this one plant. So the approach we take in the lab is to first discover that pathway.
Yeah.
So what we did is we compare kava with the related plants, for example, black pepper, to look for those genes that are only present in the kava.
Right.
And then we express the proteins that are encoded by those genes and their enzymes. And then we try to figure out what specific activities these enzymes have. So once we have that, then we have a pathway that we can then reassemble that pathway in a different organism.
I see.
So this is really the concept of synthetic biology.
Okay.
So because we understand the ground truths of how the plant making this, we take the right genes, putting all these in a different plant. So we now have a toy tobacco plant in the lab produce kava lactones.
I see.
We also have succeeded making this transplantation in E. coli, which is a bacteria, and in yeast, which is a fungal organism.
I see.
So these will become prototypes for massive production of any kava molecules that you like.
So you have two things here. This innovation that, first of all, things that really 20 years ago would have been very difficult. Now you can synthesize quite easily because you can identify what you need and then go forward to that. The other is, it sounds like you could have a revolution in pharmaceuticals because you are able, if I understand this, to produce medications that are safer. And pain, for example, it's just a huge problem and affects many people with many different conditions, and you have like a solution for that.
Yeah, so this particular study has triggered a lot of interest in the general public and also pharmaceutical industry.
Yes.
So we can think about how the pharmaceuticals are made or developed these days.
Yes.
Right? So if we have a known mechanism, we know there's a mutation in a protein in humans, like kinase, cause cancer.
Yes.
Then people start to screen chemical libraries and trying to find something that could inhibit that protein. So that works for diseases like cancer.
Yes.
But for neurological disorder, like this depression, or insomnia or anxiety, it is very hard to start from that process because there isn't a particular protein that we know would be an obvious target for regulating people's mood. And even if we start from any random synthetic molecule that does work in vitro, that means in the dishes, in the test tube, it's a very, very long way from something that's synthetic to something that would actually work.
Yes.
So that's why I think starting from medicinal plants that can already alter people's mood or have effectiveness over thousands of years of experimentation is a very legit approach.
Yes.
We already know it's effective. We know it's safe. We just need to know what's responsible for that activity.
Right.
And then develop that into a modern medicine.
Right.
Yeah, so I think it's really a shortcut towards effective medicine.
Absolutely. So you could have, it seems, greater effectiveness because you can work with a variety of plants, that it looks much safer. It just looks very interesting altogether. But the other is that I can just see this as like a whole huge field, just this. And you're right at the frontier of this.
Yeah, that's been keeping us very busy.
I bet it has, because this is not widespread yet, is it? These kinds of labs
not
like
many
yours.
labs have taken this approach towards
Exactly,
medicine.
exactly. But it, and now, unfortunately, with so many things going extinct, you have to hurry up and get things done, I suppose, identify things. But having identified the correct molecule or enzyme or whatever, does that mean that once you have that, you can, in principle, synthesize it? So even if we lose the plant that is the source, you can come up with a molecular version of this.
Yeah, you brought up a very urgent issue facing humanity.
Yeah.
We have rapidly changing climate.
yeah.
Many things we rely on for producing not just medicine, commodity chemicals, food. Those land used to plant those species may not be available, may not be there for us 20, 30 down the road, 20, 30 years down the road. So really we need new technologies to preserve. So I think synthetic biology is a way to preserve those synthetic capacities in those plants.
Right.
So then we can access the same chemical compounds from those plants without having to have the plants planted.
There, once you are able to identify these things, and then hopefully you can bring the plants back again, or whatever.
And we can also go one step further. So the excitement for me is to combine genes from different plants. And between plants and even bacteria. So we can take a plant pathway and then we implement a few genes from bacteria. So we can make so-called new-to-nature molecules that could further improve the efficacy and safety of those medications.
That is really interesting. I just want to clarify one thing. I think that you mentioned, or I read that a lot of modern pharmaceuticals concentrate on, what should I say, on like one thing, one molecule, one something or other. But in the plants and the way that you are studying things, it's multiple things. There's maybe multiple plants or multiple genes or whatever. Is that the case? That,
So in traditional medicine, traditional herbal medicine, it's oftentimes different plant materials are used in combination. And it is in very popular practice these days. I would say that the mechanism for that is very complicated. But we have also to realize that most of the diseases, we have very few cures. They're complex disease. How would you expect a single molecule to treat something that's very complicated, like neurodegeneration, Alzheimer's,
cancer, anything.
Cancer, Parkinson's? There are many things that are not working across the body, different tissues, different molecular pathways. So those traditional medicine, if they are somewhat effective, provides us a handle to learn how these symptoms are managed.
That's very.
I think that would uncover new molecular mechanism pointing to new molecular targets where the modern pharmaceutical development can go further from there.
It's very interesting in the sense that it seems like pharmaceuticals have been have taken reductionism to an extreme, whereas this says, no, it's much more complex. And so if you look in terms of complexity, you are more effective. Is that the idea?
The more we know, the more we don't know.
Yes.
So it's actually a very humbling process to understand
I bet.
how many compounds are present in even one plant, one humble plant can produce hundreds of thousands of molecules. And they interact in a very comprehensive and complicated manner with human body.
Yes.
So it'll take a long time, beyond my lab, many scientists, many years to figure out this interaction.
It just sounds like a whole new field,
It's
but
a new
not only
field,
that, a whole new perspective for science generally. It seems like we're moving toward understanding complexity a little better.
Yeah,
but
hopefully we can develop a new type of medicine.
I
for
should
those
say,
diseases.
So before you get your Nobel Prize, then what are you planning in the near future for the last minute or so that we've got here?
Yeah, so we are still very busy
I
studying
think.
many interesting medicinal plants
Yeah.
with well-proven efficacy
Yeah.
to know what molecules are there, what action mechanisms these plants are used for treating certain diseases, what are the genes that plants employ to make those molecules. And then we're taking some of the molecules, making better versions, and take them into preclinical studies. Hopefully some of these can eventually emerge to enter phased clinical trials that made into new medicine. But that's many years
It
ahead
sounds
of me.
like you've
Yeah.
got 50 years
we
of
got
work there,
started
a
somewhere.
lot.
Yeah.
But that is fascinating. And for young people starting out in this field, it would really be marvelous because you're right there at the beginning in so many ways, and with this very powerful technology to make the information accessible at last here. One more very quick question. Do you expect, I think I asked a little bit about it before, like a subset, you'll see the same molecule over and over across plants, for example, that keeps showing up. Is that possible or not?
Oh yeah, it's very possible.
Okay.
So you touched upon a phenomenon we see all the time. It's
Okay.
called independent evolution.
Oh.
So unrelated species around the globe, for whatever reasons they need to, they actually evolve the same chemical
Very
phenotype.
interesting.
So recently we're studying wound healing, the property of wound healing for certain plants. And what I'm familiar with is a medicinal plant called Paris polymorpha from the Yunnan province in China. So it's been used by Chinese people for thousands of years for treating wound. And then I was told by a friend who went to Mexico, and they found an aloe plant called Agave americana, which is unrelated to the Paris plant.
Yeah.
And I found it makes exactly the same molecule.
Very
And
interesting.
that plant is used by native people also for treating wound. So that almost indicating this particular molecule is doing something
Right.
for wound healing. So we're following that out.
That by itself is like a whole huge study, I imagine, but that's what an exciting field. And I appreciate so much you giving us this information. There's a lot more to cover. I hope we can talk with you again. This is really very informative. I wish you all the best.
Thank you so much.
Thank
It's a
you.
pleasure to be here. Thank you.